Alrighty, so look, wow, this is really a tremendous honour and a privilege, and I'm very grateful for the opportunity to come here and speak before yourselves, and always am to speak to patients and find out the things that are bothering you and ways that we can better engage both as doctors and also researchers to try to help things. I've taken a little bit of license, and I'm going to talk about AML evolving from underlying myeloproliferative neoplasm more generally. I guess, why does it happen? What is the outcome with treatment for those patients who unfortunately develop this complication, and then how can we do better? So I thought I'd just give you a little bit of background about myself. Most people don't know me here. I trained at University of Queensland Medical School, and then I did my clinical training in haematology and haematopathology at various hospitals around Brisbane. I did a research fellowship at Harvard Medical School in Boston, then came back, and as Lara mentioned, I split my time between clinical medicine at Royal Brisbane and Women's Hospital, which is really one of the biggest haematology units in the country, and also at Icon Cancer Care. And also I do laboratory research, and my lab concentrates on the myeloid blood cancers, so that includes myeloproliferative neoplasm and acute myeloid leukemia. And I'm also very fortunate to be involved in clinical trial recruitment and running clinical trials through Royal Brisbane and Women's Hospital. So the overview of my talk today is fairly simple. So what are the types of MPN, and how is the genetics of this MPN cells? Why is that important to us to understand? How does acute myeloid leukemia form from MPN? And I'm just going to talk about a couple of sort of general ideas here. Who's at risk, and what are the genetic factors that we think contributes to that? What's the outcome like for AML that evolves out of MPN, and how can we do better? So as people have spoken to you today, these are the too many diseases. So these are the myeloproliferative neoplasms are just too many of one of these different types of myeloid blood cells. So whether that be essential thrombocythemia, which is too many platelets, as you know, polycythemia vera, too many red blood cells, chronic myeloid leukemia, too many white blood cells, myelofibrosis and mastocytosis. Again, these are all the too many disorders, but they're all linked by a commonality, which is that these are derived from normal blood stem cells. These are the cells that live in our bone marrow and give rise to all of the blood cells in our body. And these disorders are linked by mutations in the hematopoietic stem cells. These are not mutations that you are born with. These are mutations that you acquire throughout life, what we call somatic mutations. And the big three are JAK2 V617F, which we know is very tightly linked with polycythemia vera, and also calreticular mutations and MPL mutations. And MPL is the receptor for thrombopoietin, which is the gene that makes our platelets grow. And so that's why it's very closely linked with ET and myelofibrosis. My research laboratory is very focused on JAK2 V617F because it is the most common myeloproliferative neoplasm oncogene. That means a gene associated with that particular condition. And it's found in almost everyone with polycythemia vera, together with the majority of patients with myelofibrosis and ET. What we do know, though, is that when these were first described, there was, I guess, a great enthusiasm in the medical community that we'd found the cause for these cancers, and really that targeting these cancers was going to be simple as it was in chronic myeloid leukemia by targeting one gene that these can all be switched off. But in fact, it turns out this is not the case. And part of that story is related to the additional genetic mutations, what we call heterogeneity. So many patients with myeloproliferative neoplasms have mutations in other genes as well. And the most common pathway is these things called epigenetic pathways. And I'll explain to you a little bit about what epigenetic pathways are otherwise. But these are genes like DNMT, EZH2, ASXL1. And these are important because not only are they associated with myeloproliferative neoplasms, they're associated with more aggressive forms of myeloproliferative neoplasms, including acute myeloid leukemia and including myelofibrosis. So what is acute myeloid leukemia, and how does it happen? So this is what we call a normal hematopoietic hierarchy, and this is I've tried to show this as somebody with polycythemia, so you can see too many red blood cells and too many platelets here. But this is the hematopoietic stem cells at the far left of screen, basically differentiating or going through a progenitor stage and then making the mature cells. And so one of the things that happens when acute myeloid leukemia happens is that a mutation forms that prevents this differentiation. So often we see early on a reduction in the normal blood cells, and that can often be an early clue that something bad is going on. And then associated with that, we can develop, patients can develop what we call pancytopenia, so a reduction in many of the lineages of mature blood cells. And then this accumulation of immature cells, which is really the hallmark of acute myeloid leukemia. And these cells may look like stem cells or progenitor cells, but they do not look like mature cells. And they give rise to infiltration of the bone marrow and the inability to make normal blood cells. And so that's what we talk about when we talk about increasing blast counts in the blood and reducing normal bone marrow, normal cell counts in the peripheral blood. What is it that causes acute myeloid leukemia to form after MPN? So there are a number of clinical risk factors and genetic risk factors. And one of the common questions that is very relevant to patients is, does the treatment that I take at this early stage make a difference to whether I'm going to develop AML or not? So the first thing I would say along these lines is that there's a small but very definite increase in all types of cancers with patients who have myeloproliferative neoplasm. And Cecily mentioned this a little bit. This is partly to do with the immune system, partly to do with the medications. But we know that all patients, all sorts of cancers, the risk is about 20% higher over the general population. So these preventative measures to try to prevent secondary cancers is very important. And certainly, as is going ahead with pap smears, mammograms, prostate checks, these sort of usual health checks to try to avoid secondary cancers. We know that when patients come in with a high circulating blast cell count, so again, seeing those immature cells in the blood is not a problem if there's just a few of them and if they're stable over time. But if we're seeing a higher level of these blast cells in the blood or an increasing level, and particularly what we think of as an accelerated phase of the disease, so often greater than 10% of the cells being these immature cells, this is something that gives us great worry as clinicians and certainly makes you think that this may be evolving into acute myeloid leukemia. We also know that those patients with very low platelet counts for some reason are at increased risk of acute myeloid leukemia. And this may, in fact, be related to the underlying genetic factors, which we'll talk about later. Myelofibrosis, being a more advanced stage disease in many cases, is also associated with a higher risk, and ET has the lowest risk of transformation to AML. But overall, the risk of transforming to AML still remains relatively low in all three conditions. And it remains the minority of patients over a prolonged period of time who will actually transform to acute myeloid leukemia. A very common question is, does treatment make a difference? And I guess we know that everything we do has both positive effects and negative effects. And so what we're trying to do as doctors is trying to balance those things for you as patients to try to make sure that we're not doing any long-term harm by controlling symptoms right now. There were certainly early studies suggesting that hydroxyurea had some increased risks, and these were what we call descriptive studies or retrospective studies. But now there's been a number of very, very large cohort studies done to look at the absolute risk of hydroxyurea and acute myeloid leukemia. And I think, I certainly believe this very strongly, and I think most doctors would, that treatment with hydroxyurea is not a risk factor for the development of acute myeloid leukemia. So this is not a drug that we are worried about giving as far as causing risks of AML. It obviously has its own side effects down the track, but hydroxyurea is not. Interferon is not associated with an increased risk of AML. And even though ruxolitinib has only been around for a few years, does not appear to be associated with an increased risk of AML. There are certain drugs that we do try to avoid, particularly in younger patients. And these particularly are drugs like radioactive phosphorus and alkylator agents, such as melphalan. And these are associated with a risk of acute myeloid leukemia that may be between 3- and 5-fold increased over baseline. But again, remembering that we're talking about an increased risk of AML over 10 to 15 years that may be at baseline as low as 2 or 3%. And so in some situations, the use of these drugs may still be necessary. And in the correct clinical context, they're very useful drugs to retain. And so if somebody comes in with very, very high counts, can't be controlled otherwise, they may be in their mid-70s or early 80s. A slight increased risk of acute myeloid leukemia in 10 to 15 years' time is sometimes an acceptable side effect in those situations. We do know that a quarter of people who develop AML from their underlying myeloproliferative neoplasm, in fact, have had no treatment, have had no cytoreductive therapy. So this is not caused exclusively by the medications, but is associated with higher risk disease, as I said, and those higher risk patients are more likely to receive treatments. So what are the genetic changes that put you at risk of acute myeloid leukemia? So this is, maybe I'll try to use the pointer here. Have we got a pointer? Here we are. So this is a kind of starting, maybe I'll start at the bottom here, and this is the chromosomes. So when you look into a cell under a microscope, you can see chromosomes when they're stained properly. And chromosomes look like these little X's. Sometimes they look like little Y's if you don't have two of the little X's, but anyway. And they're very, very tightly compacted DNA or genetic code. And that genetic code can be wound and then wound on itself again and wound on itself again. And so what we're showing here in this picture is in fact the genetic code being unwound. And basically as it's compacted in these chromosomes, which are the X-shaped things, the chromosomes are then formed of three-dimensional structures which are wrapped around what we call histones, and this chromatin pattern is the way that the DNA is held together and wound up on itself. And then as you gradually get less and less wound up, you come down to the individual bases that is in fact your genetic code up the top here. So we have genetic code sequence, so this is the T's and the A's and the C's and the G's. It's wound around these histones, which gives it the three-dimensional structure. And then these are wound around themselves again to give the chromosomes. And so we know that patients with MPN can have problems at any one of those levels, and all of those are associated in some situations with increased risks of AML. So losing a big chunk of a chromosome is never good, and there are particular chunks of chromosomes that are associated with risk of AML. So these are chromosome 5, chromosome 7, isochromosome 17, which is where a part of a chromosome is duplicated. We also know that this part here, where the genetic material is wrapped around these histones, is very important. And this is what we call epigenetics. And so the way the DNA is condensed and maintained in the right architecture is in fact very important to the way the cells use this genetic code to survive. And so this is where the changes in genes like ASXL1, EZH2D and MT3A, this is how they affect your chromosomes. And then right up to this very individual gene level is where you lose, you have changes in genes like P53. So an individual genetic code can be changed. And that actually leads, and that's the same with the JAK2 gene, for example. It's an individual letter that is changed within that genetic code. And so at any of these levels, you can develop changes which puts you at increased risk of AML. And so a group in Europe, led by Robert Kralovic a number of years ago, decided that he would have a look at patients with polycythemia vera who then went on to develop AML. And he in fact sequenced what we think of now as a reasonably small number of patients. But what he found, so 22 patients, what he found was about a third of those patients have changes in this gene called P53. Now P53 we've heard a little bit about before, and I'll tell you a little bit more about it in a second. But it's a guardian of the genome, and it's responsible to stop the cells proliferating. It does a lot of different things as well. In addition to that, of those patients who don't have P53 mutations, a very significant chunk have amplification of a part of chromosome 1 that in fact codes for a gene that switches off P53. So there's two ways that you can turn off this P53 gene, and both of them lead to leukemia, and both of them are what we call mutually exclusive. And so that means in about 50 to 60% of patients who get AML, we think this is maybe driven by loss of P53 function. And in fact, Dr. Rampal, when he was in Ross Levine's lab, showed this beautifully by function, by looking at patient samples, but also by modelling it in the test tube. And what he could show is that while many genes here, for example, the yellow and green gene, don't actually change when people develop AML, the red one here, which is P53, is dramatically enriched. So all of a sudden when the AML forms, this P53 clone takes over and becomes the dominant factor. And as I said, P53 regulates all sorts of things in the cells. So whether that's damage, so by DNA damage, what we actually mean is the ability of the cell to repair itself after damage. The ability of the cell to die when it gets a bad signal. That is important for P53. Chemotherapy response and a number of other things. So we know that P53, as was shown earlier, is a very important, so P53 loss or mutation is an important step in the development of AML. What are the other factors that are important? So it does appear to be important whether you have JAK2 or CALR or MPL. So patients with CALR have the lowest risk of acute myeloid leukemia, JAK2 in the middle, and MPL have the highest risk. But again, these are relatively low risks across a long period of time. There are also association with other genes, and we talk about these 5 main genes here: ASXL1, EZH2, SRSF2, and IDH1 and 2. And these are the genes that we look for, and we get a bit of a red flag. So if these additional genes are seen on gene sequencing, it gives us a red flag as a clinician that this is a person who needs to be watched very closely because they have a slightly higher risk than normal of running into problems with their leukemia. How can I find out about my genetic information? This is something that's really only been recently available in Australia, and again, we're very grateful for the Peter McCallum for helping us with a number of patients from Queensland here. And also, this is some so this is the Peter McCallum panel you saw before. This is a panel offered at the Mater Hospital in Brisbane by Andrew Perkins. Unfortunately, he's been poached by the Victorians, so it's no longer going to be available in Queensland. But basically, these are a way of looking at those key genes that we know are important in both the underlying cause of MPN, but also that transformation to AML. As we've spoken about, it does come with an out-of-pocket expense. They generally charge Queenslanders $600 because we're just generally more wealthy than Victorians who only have to pay $250. So the summary of it's clearly not the case. So So the summary of the first part is that there are high-risk types of MPN that increase risk of AML, but the absolute risk remains low in most cases. Some treatments, particularly the older-fashioned treatments, are associated with an increased risk of AML, but they do remain clinically useful in some situations. And like everything else in cancer, genetics are really of critical importance. So what's the outcome for patients who develop AML after MPN? I would say that the prevention of AML and the recognition of patients who are going to develop AML is really one of the absolute key factors for us as doctors, and it's something we're trying desperately to get better at, but we are still not good at identifying everybody. The survival is very low, only about 10 to 15% at 2 years after the development of AML. And the reason for this is because many patients are unable to tolerate chemotherapy, but also because the chemotherapy doesn't work as well. And bone marrow transplantation, which is really the only curative option, is available only in a limited number of patients due to a number of issues, whether that be donor availability or comorbidities. Clinical trials, I believe, are the way forward in this situation. What are the outcomes after bone marrow transplant? As I said, bone marrow transplantation can be curative. We think of the overall cure in bone marrow of this condition as about 1 in 4. But that's only in patients who can achieve remission before bone marrow transplant. And the rate of relapse, even after bone marrow transplantation, is as high as 50%. If the patients are not in remission at the time of bone marrow transplant, the rate of cure is extremely low. So there are many factors, though, that influence outcome after transplant. And I guess I saw this is a paper that was just published today, and I managed to just put it in my slide box earlier this morning. But the idea of showing you this picture is to say that it is an art form. It does take into account a lot of different factors, including patient-derived factors, how sick the patient is, what are the actual chromosomes underlying the leukemia, has the leukemia already responded to chemotherapy or not, And these are all percentages, and they all give us an idea of how the patient is going to do in the long term. But I would say to you that survival is a binary function. So if you think about computers, everything's a 0 or a 1. And that is what patient survival is all about. It's a 0 or a 1 for you as an individual patient. And the idea of saying you have a 70% or a 25% or a 10% survival chance is absolute nonsense. So that only works when we're talking about a very large cohort of patients. For every individual patient, it can only be a 0 or a 1. So how can we do better? I figure that acute myeloid leukemia after MPN is a slightly depressing topic. It's a very hard topic to talk about because we don't have the answers. But what I will tell you is that 10 to 15 years ago, when I started my hematology training, many types of blood cancers that we now think of as routinely curable were not treatable in any case. And these patients, I still remember patients dying during bone marrow transplantation for chronic myeloid leukemia. And that was 10 years ago, 15 years ago. And the difference is research, and all the big changes that have come in this field have come through research. And I mean both basic science research and also clinical research. So I'll give you some idea about clinical trials because I think this is the best way to get access to new drugs. We do not have, as far as I can tell, a single trial in Australia that specifically targets AML after MPN. But we do have a number of trials that allow patients who do develop AML after MPN to go in to these. And so, for example, at the Royal Brisbane, we have a trial with decitabine plus an antibody against leukemia cells. There's an LSD1 inhibitor at Peter MacCallum. There's other demethylating agent trials. And there's approximately 100 trials listed internationally if you go into the clinical trials register. Internationally, there's a large number of trials with ruxolitinib plus, and you can insert whatever you want there. And unfortunately, at the moment, we don't have a strong feeling that any of these trials are more beneficial than ruxolitinib plus themselves. And Dr. Messer mentioned imetelstat before, which is actually a drug we're very interested in the context of AML. And this is a telomerase inhibitor. I won't talk about it further here. So I will mention this is the type of trials that we would like to do in Australia. This is the type of trials that we should be doing in Australia. So this is a trial, and I'm grateful for Dr. Rampal for sharing the slides with me. And he's looked at ruxolitinib decitabine in patients with AML arising from MPN. And the interesting thing about this is that this is a study that's based on basic scientific research that Dr. Rampal and Dr. Levine had done in the lab to identify p53 mutations, to treat cells in the lab with these drugs both individually and in combination, and then to roll that out into a phase 1 clinical trial with a dose escalation format like this. And what is really quite striking is you can see here they're getting around about 50 to 60% of patients who are actually responding to this treatment, which I would say to you is a rationally designed treatment based on understanding the fundamental biology of how these leukemias form from underlying MPNs. And so I think this is a type of very exciting and very, science-driven trial that we should be doing to try to improve studies and to try to improve outcomes for patients in these diseases. What about laboratory research? Where does it fit in? I notice we haven't talked a lot about laboratory research today, but I think it's fundamental to improving outcomes for you as patients, and I think you need to understand that there's a role as a patient and as an advocate for patients to understand research and where it fits in. So this is something that we're doing in the laboratory, and I'm just going to step back here and put on my research hat. And so what we're doing is we're taking primary cells and we've engineered these cells to express JAK2 V617F. And what we actually are doing is we're using a viral particle called a lentivirus to bring into these cells a genetic code and a technology called CRISPR-Cas9. And what that does is it actually gets into the cell and it engineers the cell by cutting the DNA and repairing the DNA in such a way that rather than having a functional gene, such as DNMT in this case, we actually lose it. And then when it's repaired in an alternate manner. And what we can do then is actually study these cells side by side with the cells that have normal gene function. And in this situation, when we knock out DNMT3A in the JAK2 cells, we get a lethal myelofibrosis, whereas previously we only see polycythemia vera. And so we are now able to harvest these cells and say, how do they work differently? We can do sequencing on these cells. We can say that, in fact, these cells are very different when you lose DNMT3A, and that there's a number of genes that changes. We can look at the pathways that change, and we can understand what are the genes that are turned on in the transition to myelofibrosis. What are the pathways that are changed by this? And are they regulated by the underlying processes, so in this case DNA methylation? And we can correlate all of this and then go back to our patient samples and say, do we see exactly the same thing or not? And so now we've been able to actually engineer a number of different models in the lab, including EZH2, ASXL1, DNMT3A, TET2, all those bad genes you saw before. We can now genetically engineer these based on the JAK2 mutation and say specifically what happens when we combine these genetic mutations with the JAK2 mutations. How does that change the progression to AML or myelofibrosis? And how can we institute the right therapies to turn that back off and to improve survival? And so this is a grant that's been supported by Cure Cancer Australia, which is a great organisation that supports basic science research that makes a difference to patients. How can you do more as a patient and an advocate? So I think obviously promoting awareness and understanding of MPN and supporting research into MPN is fundamental. And this is both at the clinical trial levels, but also basic scientific research, because I would say to you that in MPN, more so than almost any other disease, all of the major fundamental breakthroughs and changes to clinical management have come as a direct result of basic science research, and they've all been there in the last 10 to 15 years. And so remember, JAK2 V617F was first described in 2005, and everything has happened since then, basically. We need to lobby government, NHMRC in particular. Most people would not be aware that the government has not increased NHMRC funding in about 6 or 7 years, and because of that, the NHMRC success rate has dropped from 24% when I returned from Boston to currently about 10 to 11%. So that means of all the good ideas that scientists have in the country, the government thinks they will fund around about 1 in 10. And we need to lobby non-government organisations, for example, the Leukemia Foundation in your particular situation, to make sure they are supporting research and to make sure they are supporting the future leaders in Australian haematology to be advocates for yourself as well. So at that point, I'm going to stop. I usually take questions, but obviously we'll leave them to the end. But I do want to acknowledge that I have a really amazing research laboratory who support me, and I have a lot of great people around me, and I also want to thank the patients and clinicians at Royal Brisbane and Women's Hospital and ICON for participating in all this work. Thanks very much.